Hello AP Biology students, we're going to go over the second section in Unit 5. In this section, we're going to talk about the gentleman that was on the cover photo for this unit, Gregor Mendel. Before we dive into the content here, I want to remind anyone who's watching that I only have my students take the notes from any background that's white. Any of the slides that have a black background I like to talk about but not have the students take notes on. We're going to cover Gregor Mendel's life and how he came to his discoveries and understanding of inheritance. And even though I find it interesting and I think it will help them understand the content, I don't really want them to memorize all these different facts about his life. I want my students to understand the importance of Gregor Mendel's data and discovery. Before Gregor Mendel's discoveries, there was something called the blending hypothesis. This is where, like the name implies, there would be a blend of the two parents' phenotypes in the offspring. So you can see we have this sperm and egg cell, and each of these bars represents a different phenotype. The blending hypothesis states that the zygote that's going to be formed through fertilization is going to be a blend of these phenotypes. You can see each of these bars are about halfway in the middle between the parents' bars. What this diagram is showing is a blending of the two parents' phenotypes in the offspring. Now, there was some evidence that supported this. Sometimes a red and white flower produced a pink flower. However, there was also some evidence that disproved this. Sometimes there were two pink flowers that were crossed and they got red and white flowers. So along comes Gregor Mendel, who is a monk, and he studied math and botany at the University of Vienna. The importance of this is to show that he had a scientific background. He was even a substitute natural science teacher at a local high school. His experiments took place in the 1860s. So at this time, we're still figuring out about cells. We don't know a lot about them yet, and we don't really know anything about DNA. So what Mendel set out to do was to try to figure out why certain traits were being passed from parent to offspring. He was one of the first scientists to really apply math to biology. He also followed the scientific method very closely. Mendel chose to work with pea plants because of their short generation time and easily identifiable characteristics. What this means was he could cross or he'd have two pea plants mate with each other and get offspring very quickly. And it was easy to identify their characteristics. For example, tall versus short, or purple flowers versus white flowers. Now, he worked with pea plants for 8 years and had over 28,000 of them. So to say that Mendel was obsessed with pea plants and getting data is an understatement. You can see some of the characteristics here. There was different types of seeds, flowers, pods, and stems. Now, Mendel actually kind of got lucky here because there were really only two variations of each type of characteristic. A lot of other plants are going to have more than just two, so Mendel was a little lucky. What he actually did was he took like paintbrushes and he would take the pollen from one plant and fertilize the carpel, the female part of another pea plant. This allowed him to control which pea plants mated with which pea plants. I know it sounds weird. Yes, these pea plants do go through sexual reproduction. It's obviously very different than, say, how humans go through sexual reproduction, but it's still a type of sexual reproduction. Now, one of the first things he did in his experiment was he made sure that plants were something called true breeding. What that means is he would cross two of the same type of plant again and again and again until he only got that type of plant. For example, he kept on crossing tall pea plants with more tall pea plants until he only got tall pea plants in the offspring. He would also do that with dwarf or short pea plants. He would just keep on crossing and crossing until he only got dwarf offspring. So what he did was he thought, hey, if the blending hypothesis was correct, plants should be a mix of the two traits. So if I cross a tall pea plant with a short pea plant, all of the offspring should be medium. And if I cross these two mediums with each other, then all of their offspring are going to be medium. And like you can tell, this is not what he found. The first generation we're going to say is the parent generation. That's the pea generation. These are Mendel's true breeding plants. So again, he crossed a tall plant with a short plant. The F1 generation is the offspring of the pea generation, and they were all tall. That didn't make sense to Mendel. If the blending hypothesis is true, then all of these should be median height, and they're not. They're all tall. So immediately here, the blending hypothesis is disproved. However, Mendel thought, what if I cross two of these tall pea plants together? And that's what he did. This is a form of self-fertilization, but what he saw was 3/4 of the F2 generation was tall, and then 1/4 was short. This perplexed Mendel. He didn't understand why he saw this 3-to-1 ratio of tall to short. Where did this short phenotype go in the F1 generation? And he didn't just do this experiment a couple times. I know it's kind of hard to see here, but this is his data. He did hundreds and hundreds of these experiments and came to that 3-to-1 ratio almost every single time. It's not perfectly 3-to-1, but it's very darn close to 3-to-1. And for a monk living in the 1860s, following the scientific method the best that he could, these are crazy numbers. You have to understand, this is when science was still gaining traction and we were still trying to figure out how to properly conduct science and take data. And yes, there are rumors that his data may have been fudged maybe once or twice here and there, but we can go back and reconduct these experiments and get these same results. So now Mendel didn't fully understand Punnett squares at this time or how genes were passed from parent to offspring. But he did realize the blending hypothesis was not true, so he proposed something called the particulate theory. His idea was that some particulate, he didn't understand genes yet, he just called it a particulate, was being passed from parent to offspring. And this particulate was responsible in some way for determining the genes or the phenotype of the offspring. Now, he published his work in a Society of Natural Science journal, and it was called Experiments on Plant Hybridization. And you would think that he gained a lot of notoriety, became famous, but no one really cared. The experiment was just some pea plants and some data, and people just didn't think it was that important. His last words were, "My time will come." He knew and understood how important his data was to just science in general. It turned out to be so important that we label Gregor Mendel as the father of modern day genetics. So what do we need to understand from his work? We need to understand something called the law of dominance. This is when an organism has two different alleles for a trait, and one of those alleles is the dominant allele. This will hide the effect of the other allele, the recessive allele. In this example, you can see the big A allele produces red cherries while the little a allele produces yellow cherries. You can see the heterozygous form still is red. Whenever there's a dominant allele here, the dominant phenotype will be produced. This is a perfect meme to show you what dominant versus recessive alleles look like. Next is the law of segregation, which states that during the formation of gametes, the two copies of each gene or each allele are going to separate from each other during meiosis. So in this case, each gamete will only get one of the two alleles for a genotype. This is showing that the parent's genotype is big T, little t, but they're only going to give one of those little t's to the offspring. That segregation is due to meiosis. Again, perfect meme. And lastly, the law of independent assortment. This is where genes for different traits are going to be passed to the offspring independently of each other. We learned about this in meiosis from the previous unit. This concept is really going to be important when we go over dihybrid crosses. This is a mid meme. It's just tough to find a meme for each one, but allele, I'm going to that cell. Other genes, they're fine with it. Again, independent assortment. Lastly, can somebody ask Rainn Wilson if he's related to Gregor Mendel? Because, I mean, come on. There has to be some relation there.